Hello, everyone, and thank you all for attending today's exciting session on global opportunities in digital health and network medicine. My name is Sunil Rajput, and I'm a scientist and senior business partner for health at Alberta Innovates. I will be moderating today's session. For those of you who are not familiar, Alberta Innovates is a provincial crown corporation created to support research and innovation activities for social, environmental, and economic benefits. We provide business and technical expertise, applied research services, several funding programs, and help build connections. Our scope encompasses the innovation journey from early stage discovery to commercialization and end use of knowledge, technologies, as well as policy changes. Now, before we begin, let's cover a few housekeeping items. At the bottom of your screen are engagement tools that you can use. All of the engagement tools are resizable and movable, so feel free to move them around to optimize your experience. You can expand or maximize your slide area to full screen by clicking on the arrows in the top right corner. If you have any questions during the webcast, you can submit them through the Q&A tool. We will have approximately 20 minutes for questions after the presentation, so I encourage you to use this opportunity to engage and ask questions. Of course, we'll try to answer as many questions as we can. The group chat box is a useful tool as well to connect with other participants. I encourage you all to use the tool to share who you are and where you're connecting from, but please don't let it distract you from the presentation. For best viewing experience, we recommend closing any programs or browser sessions running in the background that could cause issues. You can find additional answers to some common technical issues located in the Help tool at the bottom of your screen. Now, I'd like to welcome everyone again to Alberta Innovates Global Opportunities in Digital Health and Network Medicine session. I am pleased that we have approximately 345 registrants at this event. I was just talking with Dr. Loscalzo here. We would love to see everyone live in the audience, but we're really happy to join virtually, so this is great. Now, to set the stage, a simple definition of digital health is the integration of emerging computer-enhanced methods and technologies with health, healthcare, and society to enhance healthcare delivery and empower citizens. Now, there's an exponential rise in the scientific interest in digital health, e-health, mobile health over the past 20 years. This is based on a Web of Science search. Now, recently, the World Health Organization has published the first evidence-based guidelines for digital health in 2019. Now, from an economic perspective, the global digital health market is large. It's estimated at over 200 billion USD in 2020 with an annual growth rate of about 18%. One approach to delivering more effective and patient-centered care is network medicine. Now, Dr. Loscalzo will get into a lot of detail in addressing this topic, but just as a brief introduction, network medicine is an emerging field that uses both systems biology and network science to understand biological interactions, disease mechanisms, and treatment strategies from an integrated perspective. Now, identifying and characterizing biological and social networks or interaction networks underpins the concept of network medicine and also advances the concept of precision health. So this brings me to our distinguished speaker for today. I am delighted to introduce Dr. Joseph Loscalzo from Harvard Medical School. Joseph is a Hershey Professor of Theory and Practice of Medicine, a Soma Weiss Medical Doctor, Distinguished Chair in Medicine at Harvard Medical School. He is also the chairman and the Department of Medicine and physician-in-chief at Brigham and Women's Hospital. He received his PhD in biochemistry and his medical degree at the University of Pennsylvania, later specializing in cardiovascular medicine. He's been recognized with an impressive list of awards for his work as a clinician scientist and an educator. He is serving or has served as an editor for several major journals, including the New England Journal of Medicine. He also serves on several board positions as well as director positions. Currently, he's serving as the director of the Accelerated Innovation and the Harvard Undiagnosed Disease Network Program. Both are supported by the NIH. He's authored or co-authored more than 1,100 scientific publications, 51 books, holds 32 patents for his work in the field of cardiovascular biology, systems biology, and recently network medicine. Now, to highlight one paper, Joseph and his co-workers published a seminal paper in Nature Review Genetics in 2011, and it's been cited over 2,200 times. He's currently the leading international author in the field. Now, with all this, Joseph informed me that he still finds time to play jazz piano and has written a medical mystery novel. I don't know, Joseph, how you get a chance to do all that. Full details of Joseph's bio and the abstract of the talk are available in your engagement tools under the resources. Now, Alberta Innovates has invited Joseph to give a talk on network medicine to inspire Canadian innovators and to inform future opportunities related to network medicine and the Alberta Innovates Accelerating Innovations into Care Concepts Program, or the ACE Concepts Program. So the ACE Concepts Program is designed to enable and accelerate early-stage innovators in their commercialization journey. The program is being delivered in a series of closed call streams that are designed to support opportunities in emerging fields of health that are highly relevant to Alberta. The first stream centers on precision health. This talk is to engage all our innovators in the communities and inform what the next opportunity may look like. So there'll be a survey link that will be sent out afterwards. So I encourage you all to provide your input and help Alberta Innovates shape the future. Now I'll turn it over to Joseph to share his insights on network medicine, a new approach to the diagnosis and treatment of disease.
Great. Well, thank you very much, Sunil, for that wonderful introduction, and welcome to all of the participants. I'm delighted to be here to share some of these ideas about network medicine and its application to newer strategies for diagnosis and therapeutics. And I hope you'll see over the course of the next hour, network medicine is intimately linked to digital health and to big data in general and its application to modern biomedicine. So I'm going to talk about two themes. The first is molecular interaction networks and their relationship to disease. And the second is how one might use network approaches for more rational drug development. And to begin, let me give you the motivation behind the evolution of this field. This motivation stems from my own experience as well as many others in biomedicine. I'm sure many of you participating in today's program, that reductionism, which of course began with the atomists in Greek philosophy, but in more recent eras with Descartes, provides increasingly limited insight into understanding complex biological systems and complex disease phenotypes. And for this reason, we need to revise how one applies reductionist principles to our work as biomedical investigators. There is a long arc of reductionism in biomedicine, and with my colleague Jeremy Green, who's a medical historian and a physician at Johns Hopkins, we touched on these principles in a piece published in the New England Journal now in 2017. And the highlight of this message is simply that there have been two paths toward understanding disease and its treatment that have a firm reductionist basis. One has been led by Osler and others, who in the late 19th century, through clinical pathologic correlation, tried to draw our attention to well-defined mechanisms, ideally a single mechanism, underlying each disease state. The second path is that was forged by Paul Ehrlich, the father of modern pharmacology, who, as a dye chemist, noticing differences in the tissue uptake of different dyes, made the case that there were unique receptors that were specific to the dyes, and therefore there should be potentially unique receptors that might be specific to chemicals added to a sick individual as a way to treat that person's disease. And this notion of one drug, one disease, one drug target really stemmed from Ehrlich's reductionist work, which of course has held us in great stead for many years. Tuberculosis is illustrated in the left panel on this slide, which highlights, again, progressive reductionism, beginning with a definition of a patient with phthisis at a time when we knew nothing about the biology of the acid-fast bacillus, moving all the way through to molecular pathogenesis, and then to evolving ecology that modulates and continues to modulate our effective therapies. Now, this notion of reductionism has moved into the 21st century as we began to explore big data sets that underlie the modern genomics. And here, of course, is a typical representation of that kind of exploration. This is a Manhattan plot. I'm sure many of you are familiar with these plots. They reflect what I will call a linear narrative of disease. Along the x-axis is listed each of the chromosomes, and along the y-axis, the probabilistic linkage between a particular locus in a chromosome and a disease of interest. And this panel is taken from the Wellcome Trust Case Control Consortium's analysis of the linkage of chromosomal loci to 7 different diseases. In this particular case, I use Crohn's disease as an example. Now, the important point to make about this kind of genome-wide association study, which was certainly all the rage beginning about 15 to 20 years ago, is that there simply isn't sufficient statistical power to look at gene-by-gene associations for most diseases. And these one-off associations, while providing some insight, have really given us very little useful information from the perspective of novel therapeutic targets, with rare exception. Now, here's another example of that last point to build on the basis for developing this field of network medicine. This is a summary of a paper from Freund's group in Seattle, in which they analyzed close to 590,000 genomes from individuals who were resilient to severe Mendelian childhood diseases. Looked at a screen of 874 genes that were associated with very specific and clearly highly penetrant monogenic Mendelian disorders, and identified several individuals, 13 adults, patients, that should have produced a disease phenotype and did not. And some of those diseases are listed here on the slide from their paper. And they concluded that these, quote, individuals are buffering the effects of these rare, highly penetrant deleterious mutations, unquote. Well, of course, that's what they showed. It's really a tautologous statement. The real question is, how did this happen? What are we missing by only focusing exclusively on these single drivers of disease? And what I'll try to convince you of is that we're missing a knowledge or a notion of the interacting parts, that most biological systems are complex systems that form molecular interaction networks. And for today's purposes, I'm going to focus almost 15 protein interaction networks. These protein interactomes really serve as the basis for understanding disease biology and for understanding therapeutic intervention sites. Newer quantitative approaches can be used to analyze these complex networks that have really only evolved in the last 20 years. And network medicine, which Sunil defined for you at the outset of the presentation, is a new field that we've helped establish, myself and my colleague Laszlo Barabasi, that applies these approaches to disease pathobiology and treatment. And I shamelessly show you our book on network medicine that was edited by Ed Silverman, who's the head of our division of network medicine here in the Department of Medicine, Laszlo Barabasi, and me, which highlights many of the principles that I'll share with you in the next few slides. So the first question is, can these molecular interaction networks define genomic context, give us an explanation for what Freund's group failed to be able to explain, and thereby give unique insight into disease pathogenesis? So the hypothesis that we put forward in this Nature Review Genetics paper that Sunil cited is that if you look at the universe of all ascertainable protein-protein interactions, and I'll give you a real-world example of those in a moment, that within that universe of this complex network of interacting proteins, there sits subnetworks that are specific to each disease. That was the hypothesis. And we then set about trying to prove that hypothesis from a real world perspective. And in a paper published now in 2015, we were able to prove that that hypothesis was correct. That in this particular study, we looked at about 13,000, almost 500 nodes or proteins, and 140,000 edges or interactions among those proteins that had been defined in an unbiased way from a variety of data sources shown on the right side of the slide, many of which evolved from Mark Vidal's seminal work using yeast-2 hybrid screening methods to begin to put together the first of these protein-protein interaction networks. And that fuzzball in the background here really reflects one of the earliest versions of this interactome that we used to study the localization in modules or subnetworks of the proteins causing that disease. And here in schematic form, I show you the localization of 3 such modules for peroxisomal disorders, rheumatoid arthritis, and multiple sclerosis. And notice they're rather separate and distinct, although there is some overlap between rheumatoid arthritis and multiple sclerosis, as you might be able to appreciate, of course, recognizing that they're both autoimmune diseases. So how do we begin to derive these disease modules that we work with so extensively? Well, here's an example from the work of Rusheng Wang in our group, in which she derived the disease modules for cerebrovascular atherosclerotic disease. The first thing we do is identify a disease phenotype of interest, and then we ascertain from the literature or from other unbiased data sources those proteins that have been associated with, either causatively or simply by genome-wide association or proteomic association, those proteins that have been associated with the disease. These are the so-called seed proteins. We then map those seed proteins, which are shown in blue here, to the protein interaction network, and we identify the connector proteins, which are shown in gray, that link those seed proteins to each other. And using a variety of statistical techniques, we convince ourselves or not that these proteins do form, these seed proteins and their connectors, which link no more than 2 seed proteins together, that these proteins comprise a subnetwork or module which is discrete and distinct from other similarly sized collections of random proteins in the network. And we use a variety of statistical methods to prove that distinction. And this leads us to defining those disease modules that we then work with to understand disease biology and therapeutic potential. So what's unique about the way we use the interactome? Well, the interactome provides the missing links or the connector proteins among the disease seed proteins that create the module. And by filling in those blanks, what one begins to see is emerging from the network a variety of pathways and molecular phenotypes that can be used to explain the pathophenotype of the disease of interest. Now, here's an example of a sort of close-up shot, again in schematic form, of disease overlap between 2 modules in the interactome. And this overlap that I highlight here is that for asthma and celiac disease. If you explore the pathways that sit at that overlapping site of these disease modules, you'll find that they comprise or contain the immune network for IgA production. Now, it will come as no surprise that these 2 diseases should have some common mechanism reflected in IgA synthesis or IgA clearance. But no one had actually made this association previously, and it became painfully obvious of its importance and relevance when we began to explore overlapping pathways. Another example, more recently published from our group with Brad Marin leading the effort, is one in which we looked at an endophenotype, endophenotypes being those molecular mechanisms that are common to many diseases like thrombosis or fibrosis or cell proliferation or inflammation. And in this case, Brad was specifically interested in potential mediators of perivascular fibrosis in a disease called pulmonary arterial hypertension, which is a devastating disease that affects largely younger folks. And what he was able to identify through this endophenotype analysis of the fibrosis module in the interactome was a novel mediator of perivascular fibrosis in the pulmonary vasculature in this disease, and that's NED9, which becomes oxidized and as a result influences collagen synthesis and perivascular fibrosis. So understanding the novel pathways that emerged from this network analysis gave us insight into recognizing potential new disease mediators and therapeutic targets, which Brad continues to pursue for NED9. There are many other examples of our success in this strategy, and in the interest of time, I can't go through them all here, but I give you 6 such examples here. Most recently, one that I'll talk a little bit more about when we talk about individual personalized interaction networks. This is the paper we just published this month in Nature Communications for hypertrophic cardiomyopathy. But as you can see, we've explored a range of diseases: preeclampsia, valvular calcification in the heart, unique mediators of asthma, and in this particular case, resistance to steroids, regulators of type 2 diabetes evolution, and so forth. So these are very powerful techniques that we and many other groups have now turned to as a guide to understanding disease and its therapies. Now let's turn a little bit away from molecular networks and touch on more phenotypic networks, because we want to bring the two of them together as this talk evolves. Now, our current definition of disease really has evolved from the work of Osler, I mentioned earlier, and others like Malpighi, and this correlation, this pathoanatomic correlation at autopsy. By definition, then, these diseases are defined by their late-stage manifestations, end-stage manifestations, usually in a single organ system. But as the clinicians in the audience will know, many distinct diseases have common phenotypes, so-called convergent phenotypes. And there are common treatments, disease co-occurrence, and genome-wide association studies, which suggest that there are common underlying mechanisms for many so-called distinct diseases. So we need to rethink how we define phenotypic disease. Here are examples of convergent cardiovascular phenotypes. Up here we see a very thick heart, a thick left ventricle. There are a variety of diseases which can cause this phenotype, not just hypertrophic cardiomyopathy, which is the genetically determined disease that I'll say more about in a moment, but also longstanding hypertension that's been untreated, aortic stenosis that's been uncorrected, and infiltrative diseases like Fabry's and Pompe's disease. Similarly, here's a microvascular thrombotic phenotype, a thrombotic microangiopathy, where there is onion skinning of the vessel, ultimately with thrombotic occlusion in small vessels. And this is seen in a variety of different diseases like malignant hypertension, thrombotic thrombocytopenic purpura, scleroderma renal crisis, and so forth. One can't distinguish among these diseases by simply looking at the conventional phenotype. You really need to know what's driving those distinctions. So recognizing the importance of trying to link the unique phenotypes to the unique genotypes, we struck on the notion of building what we call a reticulotype, from the Latin for network, which reflects the disease module of interest as expressed through differential gene expression and protein expression in a given individual with that disease. And to prove that these unique reticulotypes do give us rather specific and unique information about patient-specific disease manifestations, we performed this study with hypertrophic cardiomyopathy patients, those with the genetic familial form of this disease. We obtained ventricular tissue when these patients were undergoing muscle resection in the septum of their heart as a way to treat their obstruction to outflow, which is a common accompaniment to this disease. We looked at gene expression in those tissues and obtained donor hearts that were explanted for implantation for patients undergoing heart transplantation, but for a variety of reasons, largely because of differences in size, didn't make it into the recipient patient. And we compared those control hearts gene expression with the HCM myectomy gene expression data sets and looked at a perturbation-based correlation matrix, the details of which are in the paper that I would recommend you take a look at if you're interested in the methodology. But suffice it to say that using this approach, we were able to define a rather unique collection in these 18 patients of very different reticulotypes. These are each manifestations, and I'm sure they're intentionally a little blurry because I don't expect you to be able to look at details, but you can notice the differences in shape and density. And they reflect great differences in the differential expression of relevant genes in the disease modules of these different patients, all with the same phenotype. And Brad went on to analyze the subgroups, subclassify these reticulotypes, and he was able to identify 3 or 4 different subtypes, one of which seemed to be primarily related to fibrosis, and he was able to show that the fibrotic pathways that were enhanced, their expression was enhanced in these patients, that enhancement was correlated with the degree of interstitial collagen in the tissue samples from those patients. In addition, we found that there was a unique genetic driver of this fibrotic risk in these patients, namely JAK2, which hadn't previously been shown to be associated with HCM, and now we believe is a therapeutic target for the treatment of this disease. I'll make one other point, and that is that those of you familiar with this genetic form of heart disease will remember that it's believed to be a consequence of mutations in sarcomeric proteins. But in our study, as well as many others now over the last 10 years, we were able to identify only about 1/4 to 1/3 of patients with sarcomeric gene mutations. So again, reflecting a convergent phenotype with many different genetic determinants of that phenotype, and therefore its therapy will require understanding these differences to make the appropriate treatment most precise. Another kind of network that we think about are physiological endophenotypic networks. This brings us closer to the phenotype end of the spectrum, from genotype to reticulotype to phenotype. And because of this problem with convergent phenotypes, we need to look at what subtle distinctions there may be among them to make the best stab at crossing that divide from genotype to personalized phenotype. Here's an example of how one can do that. And this is work by Will Oldham, published a couple of years ago, in which he took over 700 patients undergoing invasive cardiopulmonary exercise testing for dyspnea of unclear etiology. Now, invasive cardiopulmonary exercise testing, many of you may not be familiar with, but it involves putting someone on a bicycle, but also measuring precisely 78 different physiological parameters in real time, including measurement of pressures in the right side of the heart through a catheter placed in the arm while the patient's, from which recordings are made while the patient's exercising, measuring oxygen consumption and so forth. Now, as it turns out, clinical cardiologists wind up using about 2 of these 78 parameters. And there's such a rich data set, we argue that there must be more information that could be extracted from it. And Rusheng Wang was able to create a network, a physiological network, from the 39 nodes that she had available to her in these data sets linked by 98 physiological connections, was able to reduce that network, eliminating collinearity, to 10 nodes and 15 edges. And this gave us very useful information in predicting a clinical outcome. As you can see here, there are 4 different clusters that could be defined on the basis of these 10 parameters. And those 10 parameters defined as subclustered were able to predict all-cause hospitalization over the course of 1,500 days following their invasive cardiopulmonary exercise test. So careful physiologically based phenotypic analysis is also critical for helping work through this linkage between genotype and phenotype, the mediator of which is the reticulotype, as I've tried to show you, the personalized interactome that now can guide our view about potential therapies. So let's turn to that part of the talk now. How do we use network approaches to guide drug development? And this is also where there's been a fair amount of innovation in the last few years from our group and others. And the motivation here is that drug repurposing can be guided by knowledge of disease modules and known drug targets as one avenue for the use of network approaches. And to begin this part of the talk, sorry for the glitch, let me remind you of a couple of interesting facts. One is that approved drugs lack precision. There are high numbers needed to treat, and while they may reach statistical significance in a phase 3 clinical trial, they really fail to help many more patients than they help. And here's I think, a powerful depiction of that point, adapted from Shork's paper in Nature, where he makes the claim that for every individual, the top highest grossing drugs grossing drugs in the US help, they fail to help between 3 and 24 individuals. It's a pretty pathetic showing, if you will, and highlights the fact that we're treating mean effects or median effects. We're not looking at extremes, and we clearly cannot tailor therapies in the way that's necessary, given the incredible phenotypic and molecular diversity that underlies these otherwise fairly common phenotypes. Here's another point you may not be familiar with, and that is that most drugs are incredibly dirty. I think everyone on this call knows that they're somewhat dirty, but this is how dirty they actually are. These are data that I plotted from an analysis by Chartier, in which they asked the following question. They took about 470 FDA-approved drugs whose targets, three-dimensional targets, were known. They were able to define the binding site for those targets, and they asked, how often is that target recapitulated in the proteome? And what they found was this very long-tailed distribution. Notice the y-axis is logarithmic, and the median value for the number of targets that these drugs have is 32. Really, and this includes monoclonal antibodies, by the way, which anyone who's done a Western blot knows may be monoclonal, but they're not monospecific. So that regulatory bodies don't require that pharma to identify all potential targets. They only require that pharma exclude targets that might have serious toxicities like QT prolongation. And to me, this means that we're missing a great opportunity for repurposing. How might we consider network strategies for repurposing? Well, to think about this particular point, let's begin with the notion that typically drug targets are studied in isolation or in a rather limited way in terms of their function in a pathway that can be analyzed or ascertained. But as I hope I've convinced you so far, the complexity of this network neighborhood within which a protein or drug target might sit can dramatically influence how modulating that drug's function, that target's function, might affect some phenotypic function. So we need to begin to think about, in greater totality, the multiple pathways in which a target might behave, Remember that from that first slide I showed you from the science paper, the real world interactome, there are about 20 edges, about 10 to 12 edges or proteins that interact with each protein in the interactome on average. And you remember when you took your first biochemistry course, the professor said that the reason proteins were as big as they are is because of the strict stereochemical requirements for the active site or the binding site for a ligand. Well, we know that's not at all true. In fact, you can make many more effective catalysts with much smaller molecules. Why proteins are as big as they are is because they interact with so many other protein species in this complex, very protein-rich intracellular network. So we really need to understand that complexity and the consequences of perturbing the function of a single target with a single drug on all of the pathways in which that target may be involved. So that's point one. Point two is that we can use network principles to guide drug repurposing, and that's based on what we call the proximity hypothesis. If this pale blue oval is a disease module within which disease genes sit and drug targets sit for that disease, adjacent to which is another drug target, the drug for which is used to treat another disease, but not the disease defined by the module, the question one can rationally ask is, could this drug be used to treat this disease? Is it proximate enough? Is the target proximate enough to the disease module to influence the module's behavior? And that was what I think was a question that we asked with reasonable equipoise. And in a paper we published a couple of years ago, led by Fei-Xiong Chung, we addressed this question using big data sets, both at the molecular level with our networks and at the population level in our collaboration with Sebastian Schneeweiss. Now, Sebastian is a pharmacoepidemiologist in our department, and he has access to two large administrative databases, the TRUVIN and Optum databases, which together contain over 220 million individuals. And we asked him to ask the following questions in that database. And we were focused in this particular case on repurposing drugs for cardiovascular disease, or in particular myocardial infarction. We asked him to explore the database for the incidence of new myocardial infarctions or acute coronary syndromes in patients who had been started on drugs whose targets were close and in some cases within the cardiovascular disease module, but used to treat other diseases like carbamazepine and hydroxychloroquine. And the other question we asked was, what should we use as comparative controls? Well, we picked drugs whose targets were nowhere near the cardiovascular disease module, but were used to treat the same diseases as the drug of interest. In this case, levetiracetam and carbamazepine for epilepsy, and leflunomide and hydroxychloroquine for rheumatoid arthritis or lupus. We looked at other pairs as well, but in the interest of time, I'm just going to show you the results of these two analyses. We brought back the results of these in silico analyses to our wet lab and also tested what we thought were potential mechanisms, as I'll show you. What we found was that in the case of carbamazepine, its use, in fact, was associated with an increased risk of myocardial infarction. In the case of hydroxychloroquine, its use was associated with a decreased risk of myocardial infarction compared to its comparative control. And given the huge number of people in these administrative databases, the cells that we analyzed contain large numbers of patients, so powerfully statistically significant. And we then brought back our observations on the hydroxychloroquine to the wet lab and showed that two of the potential targets for hydroxychloroquine, when complex to hydroxychloroquine, TLR7 and 9, were associated with reduced manifestations of endothelial inflammation, as shown in the bar graphs, in response to cytokines. And these markers are clear indicators of the extent of endothelial activation or injury that are an accompaniment to the predilection to atherogenesis. So we've gone on to use this same principle, this principle of applying applying drug repurposing through network proximity analysis to other phenotypes. In this case, Jun-Sop Song and Jane Leopold performed the same kind of analysis for vascular calcification and identified 3 drugs, 2 different classes, everolimus, temsirolimus, and pomalidomide, as drugs that could be useful in suppressing calcification in smooth muscle cells. And they were able to show that she was able to show that all 3 of these were effective in this simple assay of calcification in vitro. Now, getting back to this issue of the unbiased universe of repurposable drugs leads me to just show one slide about an ongoing project with my colleague Felice Lightstone at the Lawrence Livermore Laboratory in California. They have the Sierra system up and running, which is a near exascale computing system. It's, I think, the second fastest system here in the U.S. Exascale, of course, is 10 to the 18th CPS. And the advantage to using this system for an unbiased exploration of all possible drug-target interactions is that you can do it at incredible speed compared to even existing cloud computing approaches that industry uses. So right now, if you're a pharma company and you've got a 2 million compound library and a new target of interest, you can screen one against the other using conventional energy minimization algorithms and other associated physical-chemical algorithms. And it may take 3 to 6 months to do that screen meaningfully before you get to high-throughput experimental testing of the lead compounds. With this system, in this case, we're using about 14,000 human proteins whose tertiary structures are either known or can be imputed, and about a 2 million compound open access library from the web. We're screening one against the other, and it will take about 18 months to complete this analysis. And what we'll have is a huge matrix of drug-target interactions that we can read from. It will be openly accessible. We hope that our pharma friends and partners will use the same approach for their proprietary libraries, so we could really begin to expand the scope of currently available drugs for other diseases beside the disease of interest for which they were first developed. And I'll give you an example of how we have also gone about this same principle in the COVID era. I'll show you some data that now are just getting through a second revision for a formal publication, but we put these data up on arXiv. And what we did here was we asked the same principle. Can we use proximity or other analytical approaches to identify potentially repurposable drugs that could be used to treat SARS-CoV-2 infection? And now this gets a little more complicated because we have 3 different data sets to deal with, which overlap. The first point to note is that there are over 330 human proteins to which the 27 known SARS-CoV-2 proteins bind based on Gordon's paper from last summer. So we wanted to map those to the interactome to define a covidome, which I show you here on the left, largely expressed in the lung, but we have other variants of this covidome that reflect differential gene expression in different other target tissues like brain or heart. And then we asked the same question. These repurposing candidates must have target proteins that are within the covidome or very near it. So here's a schematic diagram of how we think about this problem. Here's the virus with its proteins. Here are the host cells interacting proteins, and then they overlap to some degree with drug target disease modules, some of which may be close enough to influence the interaction between the virus and the host. In addition to network proximity, we also developed 2 other methods, applied 2 other methods that were retrofitted to this particular question, a network diffusion strategy, and a network-based artificial intelligence strategy that we then combined to create a rank aggregation algorithm. Here you can see the combined ROC curves for this rank aggregation algorithm, which, by the way, gave complementary, each of these methods gave complementary information, which added to the power of the ranking algorithm. And using this approach, we then looked at, I personally curated the drug ranking list and chose those which we put through a high throughput assay with our colleague Rob Davey at the National Emerging Infectious Diseases Laboratory here in Boston, in which the infectivity and proliferation of live SARS-CoV-2 virus in non-human primate and human epithelial cells were tested. And here you can see the list, and you can certainly refer to the paper to see the full list. And the only point I want to make by showing you this list is that when we tested the lead compounds, the top 70 of the 7,000 that we screened in silico, the high throughput screen for SARS-CoV-2 in vitro virucidal activity gave us about a 50% success rate, which was far greater than the less than 1% hit rate for random screenings. So we believe that our methodology really does give us some guidance and can make the drug discovery process, especially for repurposing, but even for straight discovery, much more rational, efficient, less expensive, and effective. So I have just a couple of other briefer topics I want to touch on before we get into the discussion. The next is, from the perspective of drug discovery, is the placebo effect. Now, we've used network approaches to begin to understand the placebo effect molecularly. So I think all of us will recognize that the placebo effect is a real effect. Many clinicians think that it's supratentorial, meaning that it really is created out of suggestion and other cortical functions that may not necessarily be associated with true molecular mechanisms. But as I'll try to convince you, that's really not true. That we can use select reductionist strategies, as others have, to identify genes that are associated with real placebo effect. And then the question that we asked was, do these placebo seed genes comprise a module in the interactome? Is there a placebome? And we identified, and this is the work of Catherine Hall and Rusheng Wang, 28 genes from prior studies, GWAS, with a real placebo arm that link to a positive placebo effect. And they do, in fact, aggregate in a module, as I'll show you. Catherine's work began with this interesting study in which he looked at polymorphisms in the catechol O-methyltransferase gene in the Women's Genome Health Study. And remember, this enzyme is responsible for catecholamine metabolism, as you'll recall. And as it turns out, there is a minor allele, which is fairly abundant, frequency of about 0.4 in the general population, and it is a gain of function allele. What she was able to show, looking at the Women's Genome Health Study, was that if you have, and I apologize for how blurry this looks, but what she was able to show was that the minor allele was very powerful at reducing the incidence of first ever cardiovascular events or myocardial infarction. And she was able to show that among placebo-treated patients, subjects, because in the study, subjects either received placebo, aspirin, or vitamin E. And here I only show you the aspirin data. She was also able to show that having homozygosity for the minor allele was actually a more powerful predictor of reduction in first ever cardiovascular events than was either of the aspirin arms. So that this is a real physiological placebo effect. There are countless other similar effects that others have shown in other studies that involve these 27 or 28 C genes. And when Catherine and Rusheng mapped them to the interactome and used the same linker or connector strategy that I shared with you in the cerebrovascular disease module construction, they were able to come up with this aggregate of clustered proteins that comprise this unique module. And if you were to look at these proteins, some of them may seem obvious because they affect the cannabinoid pathway or the serotonin pathway, but others bear no relationship to pathways or targets that are known to affect mood or affect the likely drivers of the sensation of a positive placebo. What they were also able to show is that that the strength of the placebo effect for any one of these diseases was inversely associated with the distance of that disease module to the placebo module. So the closer the two modules were, the greater the placebo effect, which by a different analytical approach confirmed the hypothesis. So what other hypotheses can be derived from this kind of analysis? What are the implications? Well, the proximity of the placebo to the disease module is directly related to the magnitude of the placebo effect, as I said. But more important, a placebo signature could be used to eliminate patients from clinical trials in order to optimize the effect size and minimize trial size or cost. And another derivative implication is that the placebo can be drugged for specific diseases. In fact, if you look at some of the targets, some of the proteins in the placebo, they are, in fact, well-known drug targets already. We may be treating the placebo, if you will, activating it through some of our current therapeutic strategies that might be more broadly applied depending upon the proximity of the placebo module to the disease of interest. This last example of a strategy that I want to share with you has to do with network medicine and the food supply, or the foodome, as we call it. In a paper Laszlo and Julia Menichetti and I wrote this past year, we drew the analogy between the so-called junk DNA, which we know now contains many sequences that are highly effective at regulating non-coding RNAs and others, regulating gene expression. So this sort of dark matter of the genome turns out to be a very important and powerful regulator of gene expression. And we feel that if you look at all the small molecules in the food supply, most of them have not been studied in the conventional sense of their relationship to health or disease. These comprise what we call nutritional dark matter, for the same by analogy to the genome. There have only been about 150 molecules, small molecules, biochemicals in the food supply that have been studied with respect to their association to health or disease, like cholesterol and sugar and such. But many, many more have remained unstudied or understudied, and offer the opportunity for us to think about food as drug. Where do these small molecules dock in to the interactome? Can one use a network approach to think about natural products and their potential rational use from a biochemical and network perspective to disease therapies? Here's an example of how complex the food supply gets. Here's sweet basil. There are 2,200 chemical compounds, small molecules identified in sweet basil. There are flavor compounds, which comprise only a small fraction of these, 20 to 25% at most. There are some disease links that have been recognized, but not necessarily to very specific compounds, with a few exceptions like rosmarinic acid and eugenol, both of which are polyphenols. And there are, of course, a variety of health effects that have been ascribed to basil, but without a clear and thorough understanding of what the molecular and, I would say, network-based mechanisms are that underlie those effects. So we can think about moving from individual eating patterns to the foodome, to the interactome, to health and disease. And here's a schematic of how we like to think about this process. We begin with individual eating patterns. We decompose those dishes into chemical compounds and create a bar graph. We take a detailed dietary history so we can get an area under the curve of exposure. And then we use that individual foodome and our knowledge of the individual genome as expressed in the protein interactome to guide the influence of these, the ingestion of these small molecules on health or disease risk or disease therapy. That's the sort of holy grail of this process. So network medicine and precision therapeutics go hand in hand. I've tried to illustrate that point throughout the talk. Again, making the case that deep phenotyping is essential for getting more precise. Reticulotyping using a network template is at least as important as detailed phenotyping and deep phenotyping for us to be able to define the best therapeutic strategy for these sub-phenotypes. Now, Sunil asked me just to touch on make a couple final remarks about challenges for big data usage in network medicine and potential solutions. And I'll just list these, and we can use the listing as a way to open up the discussion as well. But, access to these large data sets, especially private patient data sets, is essential. And of course, privacy concerns are also important to be sensitive to. We've developed an international consortium of network medicine institutions, about 18 universities in Europe and about 10 here in the US. And our goal is to build the right kind of infrastructure for free sharing of data using, at least at the outset, a federated data management strategy where individual data are still maintained in private, but the local sites perform some of the analysis, and then we aggregate analyses in this federated data structure. Another important issue is curation and editing. And the point I want to make here is that it's really important that biologists and clinicians work closely together with data managers, applied mathematicians, bioinformaticists, and statisticians, because even though it is ideal that non-biologists and non-clinicians view these data in a perfectly unbiased way, the insight that the biologist has is critical for not moving the research enterprise down the wrong path. So, for example, the drug target might seem perfectly reasonable, at least theoretically, for a particular drug that could be repurposed for a new disease for which it wasn't intended. But without knowledge of the side effects of that drug or what has already been identified in the published literature from clinical practice of its potential adverse interactions with other drugs, an unguided approach to exploring that drug's potential use could be disastrous. So these are true partnerships that have to be established. I have myself a variety of personal case studies that link our research to commercialization. Cipher Medicine, in the spirit of disclosure, is a company Barabasi and I started that has as its focus personalized the identification of personal biomarkers from network analysis that guide specific therapies for autoimmune diseases currently. Foodome is a company on whose scientific board I sit that is guided by the principles I showed you in linking individual ingestion patterns to health and disease trajectories. And I've led the Boston Biomedical Innovation Center, which is one of 3 national centers for accelerated innovation, as Sunil mentioned, that the NIH has funded for the last 8 years. And it was established really to help investigators jump the chasms in technology development. And there are really 3 major ones that you're all familiar with: inadequate funding for proof of concept or validation studies, insufficient access to resources and expertise, especially around commercialization and intellectual property and market size assessment, and lack of knowledge and experience among academic investigators in general with the development process and the commercialization process. So this program has been extraordinarily successful. We've had 50 proof of concept and validation studies, which have led to 10 SBIR, STTR awards, 9 sponsored research agreements, 17 license agreements, 11 new companies with financial support. The follow-on funding's been about 25x. as you can see here in the different sources listed in the graph. So lessons learned, find common ground among potential partners and investors. Be attentive to differences of opinion, but don't let them paralyze decision-making or forward progress. And the success of small steps as proofs of concept, scientifically or commercially, really can't be underestimated. So this last data slide just shows the path to precision medicine using networks as a guide. I've tried to highlight some of those examples for you. I haven't touched on many other forms of network analysis like microbiomic interactions, the environome, epigenetic modification. These are all other types of molecular networks that create networks of networks, which themselves can be analyzed more deeply. And I'll end with this slide that highlights the people with whom I've collaborated over the years and those who've done the work I've shared with you. And I thank you for your attention.
So thanks, Joseph. On behalf of the audience, I'll give you a round of applause. Joseph and I were talking at the start of the conversation that we are missing seeing our audience. So please engage through the engagement tools. Let us know you're there. We have 345 with us. So we're excited to open up the online forum for conversation and questions. So I'll go to the Q&A box to load up the questions. But while they're coming in, maybe I can just start with my question, Joseph. So slide 45, you had listed the barriers, which I appreciated you acknowledged. And so you had listed data access, there's privacy, curation, and there's others in terms of standardization and security. So my question is, how would you triage addressing these barriers for approaching network medicine? How would you triage?
Well, there are different ways by which you could do so. It really depends upon the context. And I'll give you two examples. I also lead the Undiagnosed Diseases Network here in Boston. It's one of now 12 other sites in the U.S. with a central site at the NIH. And this is a network that focuses on a network of sites that focuses on trying to identify the determinants of very unusual phenotypes in children and adults. And these are generally rare diseases or rare presentations of common diseases. It takes the input of a variety of geneticists and genomics folks from around the country to dissect these diseases. And the only way to achieve that dissection is with free and unfettered sharing of information across these sites. So to achieve that end in this particular setting, we worked with the NIH for it to set up all of the regulatory bodies required for approval. So there's a common IRB, a common data management assessment group, and all the institutions signed on to the NIH as being the final arbiter of these elements. And it took us a year to get all of that in place. But once it was in place, it has been incredibly smooth and easy to share information, including molecular information, highly confidential patient information. So that's one example. Another example is what I alluded to with this federated data management notion. And one of our colleagues in Hamburg is a bioinformaticist, but and a computer scientist, and his interest specifically is in data security and cybersecurity in general. So he and his colleagues have been working on this notion of federated data management for about 3 or 4 years now, and are beginning to implement it in small consortia related to clinical trials as a first pass. So now we're talking about much larger data sets with large numbers of individuals, as well as terabytes of information around genomic sequence. And the goal, as I said quickly earlier, is really that the access to the real data is confined to the institution from whence it springs. But those data are analyzed locally with the same kind of approach that every institution must use for its data sets. And then once that local analysis is performed, there are modifications made to the data so that one couldn't intentionally identify an individual patient. And those modulations, or deltas that are applied to, for example, the locus of a site, those modifications are understood and kept separately and then used to recorrect the information once the analysis is completed. But it adds several, as I'm suggesting, perhaps not so clearly, several additional steps in the data flow process that really reduces but does not eliminate completely the chance of unintentional disclosure of real patient information.
Excellent. Thank you. I'll just go to the Q&A session now. So this first question comes from Aaron Gruber. So Aaron writes, thank you for an excellent talk. I have two related questions. The first one is diseases often have a trajectory in time. Has there been progress in modeling such dynamics? So there's a follow-on to that. Are there strategies for interfering effects of unobserved factors like environment, gut microbiome, etc.?
Yeah, two important questions. The first is a really powerful question. All of the data sets and networks that I showed you are really static networks obtained at a single point in time. One measurement of differential gene expression compared to a control, for example, and then mapped to the interactome. But you're right, there is a dynamic to these networks. Gene expression changes for a host of reasons, which will modify the availability of the protein targets, which will in turn modify the degree to which that protein is bound to another protein in the network. So knowing those dynamics is crucial. It's a much harder problem, and it requires knowing the at least relative concentrations of each of the proteins in the network and their relative and their binding constants, ideally, absolutely, if not relatively. And we're working on that problem using model networks in which we know all of those parameters, trying to get a sense of how dynamics can influence, how dynamic changes over time can influence biological network behavior. There are parallels to these notions. I mean, one is complex electronic circuits, where there are dynamics. You can perturb nodes, which in this case could be some electronic element, a resistor, a capacitor, and get some information about the flow of electricity through the network. Here we're thinking about the flow of molecular information through the network. So in that respect, those parallels can be useful in dissecting the network. Not only are these trajectories that need to be taken into consideration, which we have not done yet, but aim to, but also there is differential tissue expression. That means that this universal network I showed you may have a variant in the liver as compared to the kidney. Just like the covidome was about 80% similar in the heart, the lung, and the brain, but there were 20% differences in the genes that were in proteins that were expressed in those organs. So knowing that differential expression is important. We published a paper now about 6 or so years ago, led by Maxime Kitsack, in which we asked the question, this is a question only a physicist could ask to a biologist. Laszlo said, "Why don't you get asthma in the brain?" And it's one of those deceptive questions that seems ridiculous at first, but it has an interesting undercurrent. And the simple answer is, well, the genes that are associated with asthma are not expressed in the brain. But that's not true. Most of the primary disease-associated genes are expressed in organs in which there's no manifestation of the disease. But what's not expressed, and this is where networks come in really powerfully, are the other binding partners in the vicinity of that protein in the network. So you have to have a complete or a near complete constellation of the pathways through which these primary disease drivers work to have the full phenotypic manifestations of the disease in a given organ. I've gone on there, I'm sorry.
No, that's okay, that's okay. I guess, did you cover Aaron's second part of the question? Are there strategies for interfering effects of unobserved factors?
Oh, yes. Yes, right. So I just briefly touched on that in the last slide, that there are these other networks of networks one of the first of these that we analyzed was in our first disease-associated network paper in pulmonary hypertension now in 2000, I want to say 9 or 10. In that paper, we looked at the microRNA network and mapped it to the protein-protein interaction network. And we're able to identify unique microRNAs that regulate or were associated with, as a consequence of regulation, the pulmonary hypertension phenotype, and identified microRNA 21, which hadn't previously been shown to be associated with the disease. We have an ongoing project now that is looking at the microbiome, the network of in this case, of biochemical intermediates that are modified by the microbiome in the gut, and their influence on the course of response to therapies for certain cancers. So there are environmental, environomic modulators. There are gene regulatory modulators. There are networks of networks, which we, an example of which, and a theoretical analysis of which we just published last year in Nature Communications, which addresses how these cross-network regulatory features can help mitigate dynamic changes, very wide changes in biological noise during the course of the network's operation.
Excellent. So the next question is, who is funding all the work to get a federated data management model, and who should be responsible for providing these operational costs?
Yeah, that's a really critical question. So my colleague in Hamburg, the German government's actually funding it. And then in addition, in the consortia in which his federated strategy is being applied, the consortia are funding it. In that case, they could be drug companies who are funding a clinical trial with multiple sites, or they could be funding agencies. In the U.S., there hasn't been a well-defined source of funding for this kind of approach yet, except through pharma. But it's such a critical issue for so much of what we do with human data of all sorts that it really does need to be taken on as a major, I think, central project by the federal government. Too early, though.
So I have a follow-up question that came in. Said, could you elaborate on the federated data management model? So how would network models be aggregated or collected from data sources?
Right. So, so this is always a sticky issue. Sometimes I'm happy to work in your consortium, but you've got to come to my institution to work on our computer so that you can we can maintain privacy and confidentiality. So that all of that is still is maintained around the individual patient data sets. But the data sets that are transferred once they've been analyzed in the way that I described and in other ways are transferred to a central server in which they're all aggregated from all sites. Everyone has access to that central server, as does the central team of statisticians and analysts. And that serves as the overall database for drawing inference from the analysis.
Great. I've got two drug-related questions here. So the first one is, developing new targets for old drugs is expensive and may not offer the same commercial opportunity. Generics are available, for example. How can innovators overcome this barrier?
Yeah, so, well, I don't know that developing using old drugs for new targets is necessarily that expensive. It's certainly less expensive than developing new drugs for targets, but it does cost money if that's the implication of your question, and it isn't as lucrative for those who are doing the development. So how do you make it more attractive? And, this is where there isn't I don't have a simple answer to that question. Is it a new business model that we should be contemplating? Should there be companies that focus specifically on repurposing? Is there a strategy for innovation that could be used to encourage their growth and development? Should it be commoditized? Should the government, the Canadian or the U.S. government, provide the infrastructure needed to do this kind of analysis? Should it be operationalized through a central computing facility like the Livermore facility? I think this is where people with more of a business mind than I have could, I think, make some headway, because it's not going to be solved using the existing, and I think this is the key point to your question, that challenge won't be solved using the existing structures for drug development and reward that exist in pharma now. And I can tell you, I've had lots of talks with pharma over the years around the drug development, and as soon as I get to the repurposing issue, everybody's eyes close. Not quite that rudely, but the principle's the same. So it's but I will say that there are two ways that you could make money from this. And one is that just by showing that a drug could be repurposed doesn't mean there couldn't be a better drug that could be used for that same target derived from that same family. So there is opportunity for composition of matter innovation based on a repurposing analysis. So that's point one. Point two is that I think we also need to use to come up with a better way to look at drug combinations. I didn't have time to go into this, but Vlad Elgart, who works with me, has been focusing specifically on this issue. How can you rationally look at combinations of drugs? And could you use a repurposed drug with a new drug to, if it were rational based on the targets and their relationship in the module? Is there a way to get extra mileage out of an old drug for an old therapy, extend its patent life by repurposing another drug? I think you have to keep sort of an open mind about this and not simply close the door because the word repurposing is used and it's not as lucrative as conventional drug development. And I make one last point that, I don't want to sound like an Elmer Gantry here. Repurposing is not as simple as it seems, right? The assumption is because these drugs have been in people already that we know their toxicities. Well, yeah, maybe so. But we don't know whether there'll be new toxicities that surface once the drug's given to somebody with a different disease. That's never, in patients with that disease, the drug's never been used previously. We don't know whether, for example, in the case of COVID-19, that maybe there's some drug by bug interaction that is a totally novel one. You'd only know that by a clinical trial. So what repurposing gets you is it gets you away from the need to do preclinical studies in animals, which is a big expense reduction. And you still have to go into a clinical trial, even a modest size one, to at least eliminate the potential for undisclosed toxicities. I think that's.
So that segues into the next question here, which is, have you explored the impact of introducing new side effects when repurposing medications to target different disease states than what the medication is originally designed for? So this is, I guess, this is a question directly. So I believe this question is to, in your experience, have you explored the impact of introducing a new side effect when repurposing a medication to target different disease states? So,
no, we haven't yet, but it's theoretically possible for the reasons that the questioner is suggesting and I discussed. Yeah.
Excellent. So I'll move to a different question here. So the audience member thanks you for your exciting talk. This is Edwin Wang from the University of Calgary. I can remember for reading the Food Network papers, he comments that it's interesting. The question is, we are pushing towards modeling networks in single cells, for example, networks for each cell type in the lung organ. That is, there are a couple of cell types in the lung and linked to lung disease. So I'd like to know your comments and suggestions. So I guess the question.
I think I understand what Edwin's getting at. It gets to the increasing levels of complexity when you move up from a single cell type to a couple of heterologous cell types in co-culture to an organ to a person. And you're right. A good example of that is COVID-19. So if you look at that list of curated drugs, you'll see, just as others have shown, that hydroxychloroquine comes out on near the top. But we know that it has cardiovascular toxicities which would not have been discerned based on an epithelial cell screen, either a non-human primate or human epithelial cell screen. So one needs to know, or at least consider, the toxicities of these drugs in other organs before they're used in a clinical trial. That was the problem with hydroxychloroquine. Everyone knew about its potential for QT prolongation, which, by the way, is another powerful use of this unbiased matrix of drug-target interactions that we're working to develop with Felice Lightstone, that if a drug doesn't strike at a target that influences the QT interval, for example, you don't need to go through the expensive and time-consuming process of excluding QT prolongation, at least theoretically. So there's some added value for negating potential toxicities that you don't need to pursue further from that kind of analysis.
Excellent. So while the questions are coming in, I'll just add one of my questions here. So as I was thinking about your very comprehensive and excellent talk, I was thinking about how would you comment on what would be the most urgent need right now to move forward with? So I know you've established a lot of work in this field, but I'm trying to think about what would be the opportunity for Canada to take action now.
Well, I think having a catalog of a comprehensive catalog of tissue-specific and cell type-specific protein interaction networks, that would be one. A second would be to begin to build the network of networks which are tissue-specific, namely the transcriptional regulatory network, the non-coding RNA networks that are tissue-specific, mapped to the protein-protein interaction networks. That would be two. A third would be to look at post-translational modification, which I didn't touch on here, of proteins and their consequences for network architecture and structure. That would be three. A fourth would be to, I didn't mention this, but we touch on it in a few of our papers, that the interaction network that we use now is only about, from our best estimates, about 25 to 30% complete. So further work needs to be done on identifying more protein-protein interactions, complexes with increasingly sensitive methods that can be used to build out the networks to their completeness. A fifth would be to determine whether or not you can reduce these complex systems to something that's more tractable, not to a single pathway or a single node, but to something that's experimentally tractable. That's true in both health and disease. Now, from the basic science perspective, even more fundamental science perspective, there are really three critical missing pieces here that we sort of assume and take for granted. One is that we don't really know what the intracellular concentrations are of small molecules or of proteins. That we take a liver, homogenize it, determine the concentration after purification of a protein of interest, and then back calculate what its concentration was. That's all nonsense, especially given how densely packed the cytosolic environment is with protein. It's at 0.3, 30 volumes percent protein. So knowing what the true intracellular concentrations of each of the proteins in the network is, A. B, knowing what the small molecule concentrations are that interact with those proteins, the metabolites. C, knowing what the binding constants are for those protein associations, and the kinetic constants for the enzyme activities of those which are enzymes in situ. We and other groups have looked at some of those, not comprehensive, but by illustration demonstrated first that small molecules don't diffuse according to Einstein's equation in the cytosol. If you just add a small molecule like the Hex dye, which binds to the nucleus, it's got a molecular weight of about 300, and you take fluorescence emission snapshots every 30 seconds, if the diffusion coefficient that you calculate for its size were realistic in vivo, within the first image, within 30 seconds, you should see saturation. It takes 30 minutes to see saturation. And the reason for that is that there's anomalous diffusion with time-dependent changes in the diffusion constant, which we recently showed in 2019, that exist for these small molecules with a large capacity, low affinity pool of interacting proteins, which can interfere with access of the small molecule to its real target. Schlatter and colleagues in California looked at the difference between in situ catalytic efficiency of an engineered enzyme and the catalytic efficiency of the enzyme in a lysed cell and in a recombinant setting. And there was a 1 to 2 order of magnitude difference in the catalytic efficiency. It was 1 to 2 orders of magnitude less efficient in situ than it is in the pure enzyme state. So we're kidding ourselves trying to draw conclusions or inferences from sort of the conventional biochemistry from the 1930s in governing, in thinking about the dynamics of these systems. And we need much more useful in situ information.
Excellent. So we'll take one more question, then we'll wrap up. So the question here is, do you think that there's a future for network analysis of multiple organs using exosomal RNA/proteins in serum?
Yeah. Well, I think you could. I think first of all that there is useful information in exosomes. It would be interesting to take proteins from exosomes that have been well characterized and determine whether they map to the interactome and form an aggregate or not. It would be useful to look at ascertainable protein-protein interactions within the exosome and begin to build an exosome-like network. I agree. And then it would also be useful to explore the consequences of those local intra-exosomal interactions on the function of proteins whose activities you can measure as a consequence of these exosome-based interactions. So, yeah.
Excellent. I think just in the interest of time, I'd just like to wrap up and close off our question period. I'd like to first start off by thanking our speaker, Dr. Joseph Loscalzo, for an excellent comprehensive overview of network medicine in about 60 minutes. I thank all the audience members for attending today's session. Again, I'd like to see all of your faces, but I trust that you're joining in virtually, so I thank you all for attending. I hope the discussion sparks some new ideas. This session will be recorded, has been recorded, and will be posted on our InVentures Unbound website in the next week or so. We will be sending out a short 5-question survey after the session, so I really encourage you all to engage and to provide your feedback. This will help Alberta Innovates inform future opportunities in the area of network medicine, perhaps, and advance the Accelerating Innovations into Care Concepts program. Again, thank you very much, Joseph, and we really appreciate you as supporting Canadian innovators.
My pleasure. Thank you so much for having me. Enjoy the rest of the day.
Thank you all. All right. Have a good day.